All right, in this one, I'm going to start going over chapter 5, which is based off of the 5 macromolecules. And then starting this one off, really you want to remember what is the point of these 4 macromolecules? Why did we start off this whole year with this? And it's really because life is going to be based off of all 4 of these. So really there's going to be 4 types that we have covered and hopefully you have remembered. And that's going to be carbohydrates, lipids, proteins, and these nucleic acids. And in terms of what you should know is with each one kind of related to a chapter, right? So carbohydrates, that's typically going to be used to make your, that's how you get ATP, but you got to remember that plants make them in terms of photosynthesis, and it gets broken down in cell respiration. So know kind of the function of each one of these. Lipids make up your cell membrane. And then proteins are going to be basically pretty much everything we discuss in terms of enzymes, cell signaling. Proteins are going to be tested on a lot. And then nucleic acids, that's going to be your DNA and your RNA. But regardless of the macromolecules, you have to know these 2 terms between what's a polymer and what's a monomer. Polymer is like the single building block. So think of it as a polymer would be where you have multiple ones linked together, where the monomer, as the prefix mono means single, that's a single building block. So if you want to think of it from the standpoint of, you think of Legos, right? So you would say one Lego would be a monomer, but the final structure would be a polymer. It's many Legos built together. So all of these macromolecules fall underneath that same logic. So you can have carbohydrates that are going to be linked together. Typically, we don't just have one glucose molecule. There's different carbs that are going to be linked together. Things like glycogen and starch. Proteins, most of the time, you're not just going to have one amino acid functioning as a protein. You have many amino acids linked together. That's going to make up your complete protein. And then nucleic acids, your adenine, thymine, cytosine, and guanine. That makes up your DNA, obviously, RNA. So moving forward, you just want to know the main process of what's going to break them up and what's going to form them. So if you are dehydrating or hydrolyzing something, in terms of this, one of the bigger things you want to make sure you feel comfortable with is these 2 terms. So know what dehydrating means in terms of, are you making or breaking a bond? So in terms of your synthesis of this, anytime you are making a bond, you're going to be losing water. So think of that analogy I talked about a bunch in class where if you let food on a plate sit out overnight, it's lost water, and then that food has now stuck to the plate, you've actually lost water. And if you wanted to get that food off of the plate, you had to add water or hydrolyze it. So anytime you are making a bond, you're doing dehydration synthesis. If you're trying to break apart a bond, you add water to break it apart. So this is a short little animation that walks you through an actual dehydration reaction. So right here on this glucose, I'm going to lose this hydroxide and the hydrogen from over here. And this is going to actually form a bond. So I have 2 carbohydrates here. Notice I've lost water and I've made a glycosidic bond right there. So I just wanted you to see that animation where you actually lose water to make that bond. This is a better simplified picture of dehydration. So you actually have each one of these purple spheres represents a monomer. But you want to remember that if you lose a hydroxide and a hydrogen, that can actually make your new bond, okay? And for part B, it's showing that if you have this molecule and you actually wanted to break it up, you had to add water. So that's kind of a nice overview of a dehydration versus hydrolysis. Just simply put, you lose water, you make a bond, you add water, you're going to break apart a bond. So this little quick check we've done in the past, how many molecules of water are needed to completely hydrolyze a polymer that is 10 monomers long. So I only have my trackpad, so this is going to be kind of tough to do, but the answer to this would actually be if it's 10 molecules in length, so that means it's made up of 10 individual monomers, it's actually going to be 9 bonds. So you would actually have to have 9 molecules of water to break that one up. So just to kind of wrap this short intro to chapter 5 up is, I'm going to end it there. Really, this is just kind of an overview of what are the 4 macromolecules. I'll have details on each macromolecule, but this was just kind of going over the genesis of, you need to add water to break a bond up, and then you actually lose water to make a bond.